So just a brief introduction of the lab and the kind of work that we do. So my lab builds computational biology software. We build models and applications. We predominantly focus on viruses on the biology side, and we focus on typically algorithms for big data and AI on the computer science sides. We build software typically based on hypotheses of other people. So we don't usually have hypotheses that we need to build the pipelines or the code for. We collaborate with people who have questions, and we try to build those frameworks or pipelines to answer those questions. And pretty much the work that I'm going to talk about today stemmed from a single question when our collaborator approached us about 3 years ago. And the question was, can we systematically detect genomic landmarks that are predictive of phenotypes in viruses? So essentially, this is a lab that works on genome to phenome, and we're interested in being able to detect those genomic landmarks that would be able to tell us what the phenotypes of specific viruses that they're working with is. So it turns out that a lot of breakthroughs that we're seeing these days, including the sort of breakthroughs that are powering ChatGPT and Claude and business AI in general, are excellent pathways to start or attempt to answer those questions. And what I'm going to try to do today is try to discuss some of those techniques that we're using, the AI techniques that we're using, and justify or maybe show how those new tools that are based on AI, modern AI, allow us to do things that traditional bioinformatics software just simply can't do. So the reason everyone, to some extent, has this slide is to show that it's not an easy problem, right? So the vast majority of proteins in metagenomes like annotations. So it's been hypothesized the numbers are about 50 to 70% on average in any metagenome. Some metagenomes, due to the nature of the environment where they were sequenced from, can have up to 90 or above 95% of unannotated sequences. The reason why this sort of dark matter issue, viral dark matter issue persists, despite the fact that we're sequencing more and more and putting a lot of effort, and instruments are getting faster and more accurate and generate more throughput, is because of rapid evolution, horizontal gene transfer, intense selection, and many, many other reasons. But I'm going to skip that. But the implications are dire. We can't understand the ecology of viruses and how they work unless we're able to annotate those sequences. So the first thing that we had to do is build a set of tools that would allow us to do better annotations before we start to even tackle the question of how do we predict phenome from genome. So why we can't use BLAST? Well, you take a sequence, you blast it on a database. If there is no relatively similar sequence in the database, you're not going to hit anything, right? So we've talked about these sequences being under a lot of stress, evolutionary stress that forces a lot of new sequences and diversity. But there is something that is even bigger in my mind, which is the way these software work. And essentially, a core component to sequence alignment is these similarity matrices. If you've used BLOSUM, if you've used BLAST or other sequence alignment software, you probably had to pick a similarity matrix. And often one of the most common and popular ones is BLOSUM. And essentially, a similarity matrix looks something like the following. So essentially, it quantifies or tells us how alike are two amino acids. So if I look, for example, at the C, a cysteine, and I want to compare it to a P here, or to an E, it tells me that the penalty of mutating a C to an E is -4. So this is one of the worst things that evolution can do, is mutate a C to an E, because there is a very, very high score, a very high penalty, rather, that is associated with that. But the question is, should all the mutations be penalized equally? Should I always penalize the mutation of a C to an E with a penalty of -4? And to better understand this, we can think about the role of a cysteine. So cysteine can have different roles, can occur in different locations. If you take, for example, a cysteine that is located in a binding site and you mutate that, you probably just broke the function of that protein, right? Binding site is an important region in the protein. But if you take a loop that is exposed to a reagent, that's probably not an important location, right? And if you make the mutation in that location, it's probably not consequential to the function of the protein. So cysteine has also different roles. A cysteine can be important for forming disulfide bonds. If the protein folds and you have two cysteines that are close to each other, their sulfur, the sulfur on the side chains can sort of form covalent bonds that provide structure to that protein. And cysteine is also involved in metal coordination and catalytic reactions and so on and so forth. So not only the cysteine can have multiple roles, but it also can occur in multiple locations. And it just does not make sense to say, every time I'm going to see a C that mutated to an E, I'm going to penalize it with a score of minus 4. So this concept here is often referred to in linguistics as sense invariance. We have a single amino acid that can have different roles. If we talk about linguistics, I think the analogy is even clearer if we take the example of how language works. And so, one, in language, not all mutations are created equal. So if I say, for example, I actually prefer coffee to I frankly prefer coffee, the sentence carries meaning. So the mutation here or this change here is inconsequential. However, if I say, I didn't actually say that, and I mutate the actually to frankly, and I say, I didn't frankly say that, that means I did say it, but it was not frank. It was not an honest statement, right? So this is, the meaning is completely inverted. The sense invariance that I mentioned earlier on in linguistics or in language called polysemy, it means that we have one symbol, one word that has different meanings. And say, for example, the river bank flooded. I'm referring to that, the side of the river. I could say I deposited money in the bank, and bank here means a financial institution. Or I can say you can bank on her support. You can count on her support. So same word, 3 different meanings, same cysteine, 3 different functions, and mutations are not created equal. So if you've been using generative AI, chatbots lately, wonder, well, how does it know the difference between these things? And the breakthrough was in a paper that was published in 2017 called Attention is All You Need. It's a paper that was published by a group from Google. And it introduced a new neural network architecture that's called transformer. And sort of the takeaway message from that paper is that there is one way to encode in this new model architecture, the transformer, a context-dependent way to encode the value of a word. And this simple sort of proposition completely changed how we do modern natural language processing. It had immediate results from machine translation quality jumped dramatically. Question answering became genuinely helpful. And it is essentially at the core of pretty much every language model, every chatbot that you use encodes this concept of attention. So the claim is if this new neural network architecture that is powering language models can learn that bank means different things in different contexts, then we probably can get it to understand that a cysteine can have different roles and mean different things in different contexts. So just to make sure that we're all on the same page, and before we go any further, wanted to emphasize that in machine learning, typically models can't work with symbols directly. A C does not mean anything in machine learning. We have to give it some numerical representation. And so we map discrete inputs like words or amino acids into a numerical vector. And a vector, just like a vector in R, is a collection of real numbers. A vector in R or list in Python is what I'm referring to here. So I'm creating a vector in R for bank that has a bunch of numbers. And so this is the representation in the computer's memory or in the computer of what a bank means. So typically these representations are hundreds, if not thousands of numbers. But for the sake of the example on the next few slides, we're going to think about it as a vector of size 3. So what this vector essentially represents is just the word's position in 3D space. So each number here is a coordinate. So this is the coordinate in dimension 1, the coordinate in dimension 2, and the coordinate in dimension 3. And bank is my point is in this three-dimensional space. So what the attention mechanisms, going back to it, what the attention mechanism does is it makes sure that any word does not have a single representation, which was the case before transformers. Any word has a single representation. There's a unique vector that always represents that word. Post or during with the transformers, it makes sure that each word is dynamically computed based on the surrounding word. So it's going to take the context of that word in order to determine what is the vector that we can associate to that new word. And attention, you can think about it as the word, when computing its representation, it asks the other words in its context, how relevant are you to me? So it says, bank, how relevant are you of? How relevant are you to me? How relevant are you to me? How relevant are you to me? And so on and so forth. And what I'm representing here is sort of like a histogram that shows the contribution of every word, how they attend to, how they provide attention to, or they attend to that query word, which is bank. So sat provides a lot more attention than Mary. Mary, we don't care that Mary's sitting there, but the fact that she's sitting, it's maybe not banking on her support, maybe not a verb, right? And by the river, the fact that we have river and we have sat, this bank sort of is pulled a little bit more towards the riverside than financial institution or than a verb, the verb support. So imagine that we have a space that already has some words in it, and it has the words edge, riverside, shore, it has deposit, money, and vault. And if we have a couple of sentences, the first one being, Mary sat by the bank of the river, and Mary went to the bank to deposit a check. What the transformer is going to do, what the transformer architecture through the attention mechanism is going to do, is going to assign a representation to bank, the blue bank, such that it's closer to these words, and it's going to assign a representation to the bank in red so that it's closer to the other words. And we see that there is this sort of clustering of words by semantic similarity. Semantic similarity is just meaning, right? So bank here, the blue one, we got a concept of riverside and shore and edge, and by the same token, bank here represents more the concept of money and deposits and vaults and so on and so forth. So it turns out that just like we can do this with language models, we can do this with protein language models. And protein language models can do exactly the same thing, and generate what we call embedding. So these representations, I've been calling them representation. The official formal definition is an embedding. So an embedding essentially is just a vector, just like the vector we saw earlier on. They're dense in the sense that they're not sparse. They don't contain a lot of zeros. They're mostly actual numbers. And they're learned by either large language models, if we're generating embeddings for words in a language model, or by protein language models, which operate exactly the same way. One works with words and text, the other one works with proteins and amino acids. The underlying assumption when you're working with a language model and generating these embeddings is that similar inputs end up close together in vector space. Just like I showed earlier on, I said the bank, the concept of bank that is financial institution, ended up getting a vector that placed it close to things that are similar, semantically similar to it. And if we apply this to protein sequences, we get exactly the same representation. We get, for example, C is close to G because or bad example, an N and a T, they're close to each other because they're polar. We have all the hydrophobic amino acids that are clustered here, and so on and so forth. So there is this concept of similarity that we can learn in an unsupervised way by training these transformer architectures on protein sequences. So this raises the obvious question. Do we still need similarity matrices? Is there any way that we can skip this concept of similarity matrix and rely on the representation of the protein sequences? So imagine I have two protein sequences. I have sequence 1 and sequence 2. Sequence 1 is in red here, and it has a bunch of amino acids, 1, 2, 3, 4, 5, 6, 7, 8. And every amino acid is represented as a dot in two-dimensional space, right? Remember that we're typically in very high dimensional space, 1200 dimensional space. For the sake of the example, I'm showing them in two-dimensional space. And we have sequence 2, MVQITVPF, and they're also the dots here in the two-dimensional space. And these points end up occurring in this 2D space. And the question is, if I want to know what is the cost of mutating a C to a G, can I look at the distances between them and tell how far it is? So for example, what is the cost of mutating this I here, this M here to an I here? Or this M here to this I, or this M to the Q? Well, I could just look at the occurrence of the M, the blue M, and say, well, how far is the M from the I? How far is it from the Q, and so on and so forth, and compute these distances on the fly, right? Without resorting to this distance matrix that is context agnostic and does not, understand the role of the protein that I'm working with. So that's the challenge that we started to work with. The challenge is that when we say semantically similar words get semantically similar vectors, are located in the same region. This is amino acids with similar rules get similar vectors. This is the theoretical motivation or the aspiration that we want to get. It's not a guarantee, right? Things are somewhat in the same vicinity, but how do you turn this theoretical motivation into something that we can work with? Well, it turns out that we could do that. And essentially what we did is we built an algorithm that does exactly or implements exactly this idea. So given two protein sequences, I'm showing here rows show the red sequence and the columns show the blue sequence. We can compute the distance, think of it as some sort of Euclidean distance. In our case, it's not the Euclidean distance, but for the sake of the intuition, it works. And this, for example, tells me the distance between the I and the M, at the first position in the two sequences is 0.77, and so on and so forth. We take this matrix and we turn it into a hypergraph. And essentially what this hypergraph tells me is that it can allow me to visualize two types of relationships between the amino acids, what we call mutual best hits. And those are, for example, this M here. This M is closest to this M because this is the highest score that we're seeing in the matrix. And this M is closest to this M. So they're mutually each other's best hits. And we'll represent them using this green square here. And we have some yellow squares, which are cases where one is the second's best hit and the other is the first best hit. So for example, if we look at this one has the highest score at 0.9. The second best score is 0.86. And this one has the highest score is 0.86, which is this one. So it has a yellow representation between the I and the V, which is this one here. So we take this graph here, and we have this new algorithm that allows us to parse it and figure out how we can align the two sequences in a way that makes sense. So we jump from this to a representations that look like the following. So the alignment that this gives us is I'm aligning I with nothing because I does not have the first I in the red sequence does not have a match. The M aligns with the M, the M with the M, the I with the V, the Q with the Q. There's a gap here and so on and so forth. So what we just did, we essentially took that traditional sequence alignment algorithm that powers BLAST, powers MEFIT, powers alignment software in general, and we extracted that component that is the blow-sum matrix or the PAM matrix, and we replaced it by a similarity matrix that is computed directly from the embeddings. And this is a paper that was published in BMC Bioinformatics in 2024. And the results were striking in how much more sensitive this approach is compared to traditional BLAST. So we've tested it on a bunch of different data sets, and I'm just showing you one of the results here that essentially takes 13,000 VOG representative, VOG sequences from viral orthologous groups, and tries to annotate them with a threshold of 1e minus 3. BLAST is able to annotate 48 sequences that we can't annotate. We're able to annotate 5,810 sequences that BLAST can't find, and we both find about 2,783 sequences. So incredible results. And we've tried this in the lab on a bunch of data sets, the virus of rhizobia, on viral metagenomes, and each and every time the collaborators are just shocked by how much sequences we're able to find that BLAST is not able to find. We've got a fairly good precision. So precision is when we say that something, for example, is a polymerase, we're about 94% sure or accurate in that call. And if we have about, let's say, 100 polymerases in the data sets, we're able to retrieve about 98% of those. So very high precision, very high recall. We could do a little bit better here, and I'm going to talk about this in a second. So this is an actual example. Well, we have two sequences, and we BLAST them, and or we try to align them against each other. We BLAST them, and BLAST says no significant similarity found. I'm pretty sure that all of you are familiar with this screen. And what we're seeing here is the alignment using soft alignment. So every single red dot is a match between a column in the row and a column in the first sequence and a row in the other sequence. And so every one of these dots, we don't have sort of a theoretical mathematical framework to estimate e-values, but what we've done is we've tried to estimate what is the probability of observing an error. And we did this empirically. So we took about 12 million alignments where there shouldn't be any sort of matches between the sequences, and we tried to quantitatively or empirically estimate the probability of false error. And we're seeing that the probability of obtaining a single one of these dots is about 1e minus 5, right? And doing a little bit of probability in math, we realized that the probability of seeing a stretch of 3 consecutive amino acids that are aligned between the two sequences is about 3.91 e minus 29. So it's absolutely impossible to see such a stretch, complete alignment between the two sequences if it wasn't actually real. So this was fantastic, and we had a fairly okay precision, great precision 94. And the question was, can we improve? Can we further improve on that precision? So it turns out that the language model that we use, the protein language model that we use, called ESM2, it's a product of Facebook, Meta, Facebook Research, which is now Meta. And so they released a bunch of protein language models that are absolutely fantastic. The only problem is that these language models, they're foundation language models. So their foundations typically are generalists. So they're trained on data sets that are heavily dominated by cellular organisms. So essentially, the viral proteins are way underrepresented in this data set, and the model does not get to see a lot of viral sequences, so it does not understand viral speak, right? So, and we know from publications that the performance of protein language models is heavily dependent on the proportions of the sequences that it sees in that data set. So if it hasn't seen a lot of fungi sequences or a lot of viral sequences, it's not going to do very well on those types of sequences. So the solution, and typically with machine learning, you take a model, or these foundation models, they're typically fine-tuned. So when you have a generalist model, you have a generalist doctor, you can fine-tune it into a cardiologist or an endocrinologist or whatever it is. We can do the same thing, and we can, in so doing, sort of tweak it to better understand the viral characteristics or the viral speak in general. And so that was the next step, and we did that. And again, the results were staggering, were just incredible results. This was a paper that was published by one of the graduate students last year. And just doing that on viral sequences substantially improved annotations even more than we were doing with the traditional model. Better performance on orthologous groups, prediction, family, and domain level annotations. Enhanced clustering when we're clustering sequences. we're getting better results. I'm going to show you some of that a little bit later, but the clusters are very clear delineation and separation between different clusters. The alignments that we're doing with soft alignments are even better when we fine-tune the models to better understand viral sequences. And this also, this work came with a framework that pretty much allows anyone to take ESM2 and fine-tune it to whatever rare or underrepresented types of sequences they're working with. So if anyone in the room has some, I don't know, people, I'm saying it for the people who are working on fungi, we can definitely do this. And it takes 3 or 4 days to take a model that's generalist and specialize it onto something a little bit more complex. So question was, okay, can we do better? Can we do better? And this was motivated by the fact that these models, these foundational models, typically they have a limited context. They're trained to take one protein sequence at a time and compute embeddings for every amino acid in that sequence. Due to computational constraints, they truncate the sequence at about 1,000 amino acids. So they can't handle anything that is above 1,000 amino acids. So this is what we're currently doing. We're taking this amino acid. This is the same example of bank that I had earlier on, that's sort of looking at what's attendant to it, or computing this concept of attention in its context. And this is really the crux of what is making these models really good, is this concept of attention. But aren't we sort of shortchanging ourselves by looking at just one protein at a time? The reason is that viral genomes, they're not just a bag of proteins, they're coordinated systems. So a protein at one location may need to attend to protein thousands of positions away, right? So what actually is important to this is another cysteine that's located here that actually allows the structure of this complex. Imagine a cysteine that's binding a zinc finger, and the whole thing has to just fit together properly, right? So we need to be looking at attention at incomplete genomes. And this is a really, really, really challenging technical problem. Sorry, it's mostly a computational problem, and to do it actually and accurately is very challenging. So this is somewhat analogous to, and not sure if you remember, but when ChatGPT was released a few years ago, it had a fairly small context. You would type a question, or 3 or 4 questions, and says, I can't handle more questions, you got to start new sessions. That's the context. That's how much information it can handle, how much info it can sort of process at once and look for attention between these words. So now you'd say that, well, now I could just upload 100 PDFs and the contexts are massive in these things. So it turns out that they use a lot of tricks that are really good for working with words, but not very good for working with viral genomes. For instance, if you're working with a large text, the way ChatGPT or Google deals with this is by only looking for attention in the neighborhood of every query. So if I'm computing the embeddings of this amino acid, I'm only going to look a few words downstream and upstream, right? So let's say 10,000, and you have this concept of sliding window, and it never looks very, very, very far away in a systemic way. So our solution was like, well, maybe we could come up with something, and that something could leverage the biology. And we know that there is interaction between proteins. So how about we use something like that, protein-protein interactions? We know that we can figure out a way to do that. And so it turns out that we have a way of computing this. So we say attention when we're looking at the same protein, but we say cross attention when we're looking at 2 different proteins, right? So when we have these 2 proteins, we can compute cross attention. It tells me that this position amino acid here has something that's affecting it here, or that attending to it here, and this amino acid here has some other amino acid that's attending to it in a different protein. So we can compute and we can quantify cross attention, and we can know that there is a lot of cross attention between I and J. There is some attention or little attention between I and K, and there is no attention between I and L, right? Between these 2 proteins. And so this is a matrix that sort of tries to formalize the way we look for the way we look for region that we're going to compare for cross-attention, rather than use this sort of naive way that is a sliding window, like modern language models use to handle massive amounts of data, we do it in a more systematic way. And for example, if I'm in protein 1, I'm going to consider attention within protein 1. So all of the amino acids in protein 1 are going to look at each other and say, oh, how relevant are you to me? How much are you affecting me? And so on and so forth. But they're also going to do it for the sequence that's the protein that's upstream and downstream. So if I'm, for example, sequence or protein P4 is going to consider P5 and P3, right? So automatically, I have this concept of sliding window, just the new languages have. But what we add is this concept of protein-protein interaction. So knowing this or being able to predict this cross-attention, we can say that if two proteins are affecting each other, then we're going to include them in the calculation. So for example, P1 is in relationship with P4 and also with P8 and P6. So we're going to include them in this concept of cross-attention. We're going to do it. And so everything that is colored here is things where the computation is going to occur. Everything that is white is where the computation is not going to occur. We don't care about it. We're going to save space. We're going to save time by not doing it. This is, as I mentioned, a quadratic. It's a very challenging problem. If you take a sequence that has about 1,000 amino acids and you want to do full attention on that 1,000 amino acid, it takes you about 24 gigs of RAM for 1,000 amino acids. So imagine a genome, a viral genome that's, let's say, 50,000 amino acids. It's going to take you terabytes of storage, and it's going to take which is impossible. It's going to take you a cluster to compute it on a single genome, let alone on an genome. So by implementing this, again, the results were very impressive. We were able to expand the context from ESM2 that has a limited context to 1,000 amino acid to up to 61,000 amino acids on a single GPU card that costs about $3,000, a GPU card with 32 gigs of RAM. The vast majority of genomes are deposited to the NCBI, have a length that is less than 60,000 amino acids. So this covers about 98,000, 98% of the viruses on the NCBI. And so imagine you can pretty much analyze full genomes, full viral genomes on a server that runs in your lab, which is pretty cool. What was really surprising is how we were able to implement or improve actually the quality of soft alignments by doing this too. So it turns out that if you use the traditional ESM2 and you compare any two amino acids and see how similar are they. Let's say that we're comparing, and the comparison results in a score between 0 and 1. By default, those scores are constrained between about 75 and 90. They don't use the full scale or the full range from 0 to 1. So whenever I say someday, if I compare two things that are very similar, it's going to get to like 97, 98. If I compare something that is very dissimilar, it's going to be 0.71, 0.72. So we have a range between 0 and 1, yet the score, the results that we were getting were constrained between 70 and 90, and we never understood why. And this is this peak that we're seeing here, and the scores that we're getting was very, very narrow in the traditional model. By building this model and improving this model so that it takes into consideration the full range, the full genome, we end up with a lot wider and more expressive range of scores. And that pretty much improved everything that we've done with this model, from annotation to classification, to clustering, to alignments, and so on and so forth. And typically people who use these languages talk about perplexity, right? What we compare is the perplexity of the algorithm. You can think of perplexity as how surprised a model is to see a word. If you tell me a word I've never heard, I'm going to be, oh, maybe perplexed, right? Because I've never heard that language, that word. But a model that knows and is not fazed by new terminology, it's not very perplexed. And so when we compare our models, which are the two lines at the bottom, their perplexity is constant regardless of what we throw at it from these metagenomes. So it's doing substantially better at at handling these viral sequences. Okay, so all of this so far was just that introduction to discuss the tool that we need to answer the question is, can we systematically predict metagenomic landmarks that allows us to predict genome from phenome in viruses, right? And so we had to do all of this, build this model, but the question is, right now is, okay, we have parts list, we have a bunch of genes or proteins, we have some description, we have well-annotated proteins. Can we find a way to tie them together in order to come with this modules or these sort of, we didn't have a name for what we're looking for, combinations that are predictive of phenotypes, right? And so that's the question. So we decided to focus on replication mechanisms, where phenotypes that are relevant to replication in general. And the reason is because one, replication is subject to intense selection pressure with really direct phenotypic consequences, but also because our collaborator had done a lot of work on replication mechanisms. They understood them very well, and they had a lot of examples they could show us where that was in action. And so here's an example of phenotype to genotype. DNA polymerase. If you take a DNA polymerase, type 1, type A, family A DNA polymerase, and you look at position 72 in the wild type is E. coli, but if you align your sequence to the wild type and look at position 72, you can predict the infection strategy from that position. If at position 72 we have a phenylalanine, it's going to be virulent. If we have a tyrosine, it's going to be virulent. If we have a leucine, it's going to be temperate. So from a single position, you can tell what's going on. But it turns out that this was sort of a fluke. A fluke in the sense that a single position is rarely predictive of what's going on. Typically, that position is related to a module somewhere else. And when you delve a little bit deeper, you look at these, you find that there is another gene. So this is the polymerase. This is the helicase. So depending on which version of the helicase is, you have a superfamily 2, superfamily 1. Do you have a GP4 type helicase? Is the ribonucleoside reductase present or absent? That is what really tells you whether something is temperate, virulent, or not, right? And so how do we come up with these combinations? How do we come up with these sort of assortments of modules that are predictive of the phenotype? In business, there's something called market basket analysis. Market basket analysis, it's a method to discover items that frequently co-occur together. So it's a very popular approach in business analytics, and it looks across many baskets. So imagine that you have a Safeway US that looks at every single receipt, millions of receipts, and analyzes them to look at what co-occurs in the same receipt. People who bought X, what else did they buy, right? So here you have the example of the person who looks at this and the bananas and says, okay, customers who bought this item also bought this item. And so this is exactly what we're looking for. When we see X, what else do we see? And what sort of assortments do we, can we tell apart in conjunction with a specific phenotype? So let me jump to an example and natural language again, or real life example again. So you could look at market basket analysis and look at all the receipts of Safeway. You could say, oh, I'm seeing that a lot of whole grain bread, almond, oat milk, Greek yogurt, quinoa, fresh vegetables occur on the same receipts. And you could say, you know, maybe this is a basket signature in there. Statistically significantly, we can tell that if a basket contains low-fat milk, I predict that it's going to have a high probability of also containing whole grains, fresh produce, and low-sugar items. By the same token, if I look at the receipts and I see something like, let's say this one, if we have a lot of store brands, whatever the store brand for Safeway is, if we have a lot of bulk items, if we have a lot of generic products, discount items, we can build a rule. And that rule would be if a basket contains one store brand, high probability it's going to contain store brands across multiple categories, or maybe even some generic products or bulk items. So that's exactly what we're trying to do because this, in business, it's called market segmentation. It allows us to label these and say, for example, these are maybe the health conscious basket. This is the indulgence basket. They like to buy ice cream and high probability of cookies and high sugar and full fat items and so on and so forth. It allows us to put labels on these categories. And that's exactly what we're trying to do. So what we set out to do is try to find these items that co-occur together in every single virus. We have a bunch of viruses, right? Every single genome is a receipt. Every single protein is a product. And we want to find what products co-occur together, right? And essentially what we did is to build this framework. But one thing that we had to do, we can't talk about butter. We can't talk about bread because we have proteins. Those proteins have descriptions. So the first thing we need to do is find all the proteins that represent butter, or all the proteins that represent, for example, a helicase. We don't have it described as a helicase. It could be, for example, GP4, SNF1, and so on and so forth. So we had to take these embeddings, and we use the embeddings for clustering. We don't cluster on the sequence level. We don't do anything anymore at the sequence level. We only use embedding for pretty much everything that we do. These, starting with about 11,000 protein sequences, clustering them generates this massive tree that has about 1,000 manually curated. We absolutely curated. The general we, will here spend weeks staring at that tree here and trying to cluster. This is butter, this is beer, this is wine, and so on and so forth. So we end up with these clusters, and then we know which things affect which proteins, which affect which other proteins. So this is essentially the cross attention that we computed earlier on, and it's a way of saying, when I see protein 1, I also see protein 2, because there is that cross attention and this dependence between them. So using that information, we built a cluster. We did some sort of cleanup using cluster analysis, simply because it was such a massive cluster that we couldn't run market basket on it directly, and we had to split it into sort of independent components or independent sort of subclusters. And each one of these subclusters then we took and we run market basket analyses. And what we end up with is something like this. We see that, for example, this is a protein module. This protein module contains protein cluster 1, 2, 3, 4, 7, and 9. And this is another module that contains these protein. And these rows here are the genomes. These are receipt number 1, 2, 3, and so on and so forth, or genomes 1 through N. And this is genomes N plus 1 to M. And so I'm going to show you the results on a very small data set. This is about 44 genomes. And it was super, super interesting. In these 44 genomes, what we find is that this set of 24 genomes and this other set of 20 genomes, so total of 44, contained the same genes, our core functions. These are, for example, helicase, UVSW, RNRs, RDRs, DNA polymerase, helicase. These are core functions, but then each one of them sort of diversified their own way. So they all contained, for example, the ingredients to build a cake, but then one had sort of the ingredients to do icing, and the other one had ingredients to, decorate the cake some different other way. And these. So this is a lower dimensionality representation of the genes that we're observing. If it's a circle, it belongs to genotype 1. So we call the first one, we call this genotype 1. So we can't call it phenotype yet, and this is genotype 2. If it's a circle, it belongs to genotype 1. If it's an X, it's genotype 2. And every single sort of unit of a circle or an X is some sort of protein. What we're observing here is that all those observations that have the same color is a core function, right? So for example, the X and the O's here represent DNA polymerase 1. All the ones that are different between genotype 1 and genotype 2 have different colors, but are very close to each other. And this is where we're saying that the clustering was incredibly amazing in the sense that this here is called an RNA sigma, and this one is called a sigma factor, right? These are the descriptions. They occur separately. There's a lot of dissimilarity between their sequences because otherwise they would have been clustered together. They were not clustered together, and they occur close to each other. And one occurs in one genotype and the other one occurs in a different genotype. So they represent something that's similar but done differently somehow. And you see them so similar, core function, ancillary function or supplemental function. But then you have these one-off things. So for example, this one-off thing, thyrodoxin, is occurring completely separately, and RAD50, not 50, it was, YOLD-like protein is occurring separately. These are something that are unique to one genotype or the other. This is unique to genotype number 1, and this is unique to genotype number 2, right? And so, so these are the genes that are present in either genotype 1 or genotype 2, or the proteins that are there. And we set to understand, so what's the difference between genotype 1 and genotype 2? Can we come up with some sort of phenotype that is relevant and that we can use to explain what's going on? And we're not molecular biologists, but with the help of some incredibly amazing tools, ChatGPT and colleagues and collaborators, we started to dig in a little bit deeper into what these proteins do. And for example, if you look at this attention, polymerase 1 has a lot more dependence in this case on this helicase GP41, whereas the DNA polymerase here has more attention on this helicase UVSW. So in this genotype, they prefer this helicase, and this genotype, they prefer this helicase. So both helicases are present in the genome, but we have sort of differential preference across the two genotypes. So the hypothesis, looking at a few of these, the hypothesis was that, well, maybe phenotype 1, maybe genotype 1, we can associate with a phenotype where we have more emphasis on processivity and speed during DNA replication. And phenotype 2, we can associate with emphasis on accuracy during replication, right? So pretty much all the telltales that we're looking at, here's another example, all the telltales that we're looking at, DUTP in the first one linked to more rapid processivity in DNA replication, linked to more accurate DNA replication. So this is, let's do it fast and quick, and we don't care about the errors. And this is, no, let's be deliberate. Let's do it slowly, and let's replicate very nicely and make sure that there is no errors, right? So this is the market segmentation if we're doing a market basket analysis. And this is on a small subset, but then we scaled this analysis to all the genomes that we had. We ended up finding 19 different modules. The smallest of these modules had 9 proteins. The largest one has 17 proteins. We don't see them very well, but we're mapping the colors on top of these clades. We can see them slightly better here. Every single dot is a genome, and the circle above it, or the shape above it, the color above it, shows the module that maps onto those genomes. So we see that there is some reason to this madness. There is some validation that these groups of genomes have something that distinguishes them. And what is it that distinguishes them? So it's a little bit more complex in this case. We have these complex genomes, and we don't know what the function does. But digging a little bit deeper into the composition, genomic composition of these genomes, what we did that helped as well is sort of classifying these proteins as one of 5 categories, either associated with replication, RNA processing, morphogenesis, metabolism, and lysis. And what we end up with is somewhat similar stories, similar approach that we did earlier on. We end up with, for example, this module 3. We see that it has an anaerobic RNR that we know enables replication in oxygen-limited hosts. It has 2 types of helicases and topoisomerases that support genome recombination and adaptation. And the hypothesis was, after looking at some of the genes that are in there, well, maybe this module is present in that class of or market segment that has flexible host range strategy, right? By the same token, we're able to find modules are specific to the segment of oligotrophic specialists and thick-walled host specialists. So for example, this one has high fidelity replications, it has choline to penetrate thick peptidoglycan cell walls and so on and so forth. So this was surprising. So definitely a lot of validation that is needed. But what's super interesting is from this here, you can imagine that we can add metadata. So if we know, for example, that one module occurs in cold water or tropic environments or in soil, that could be a more sort of interesting way to look at these modules, but also a way to validate these modules. Okay, so I'm gonna wrap up. So the conclusions that I'd like you to hopefully leave with is classic tools are fantastic, but we're definitely in an era where AI has made huge dents in a lot of things that are relevant to us. And so perhaps adapting the classic tools to the modern power is the way to go. By doing this, we see that we've gained surprising power when building context-aware embeddings. And as I mentioned, we use embeddings for everything that we used to use sequence for. We don't use sequence anymore. We use embeddings for everything that we do, and can be applied to pretty much any species, right? We're focusing on viruses here, but we've tried to apply it to anthrax as a collaboration with the Norris group, and it worked super well. We have a paper that was published last year in Pathogens, and it again showed amazing sensitivity when blasting, annotating the sequence, and then finding these modules in anthrax. The modules is actually what we need to be thinking about when we're linking genotype to phenotype, not a single mutations, not pathways, but modules. And we're still looking at validation, and validation we think needs data. Being able to validate this in different environments, different genomes is the way to go. So this work was done predominantly by the students. Will, who's here, has done pretty much all the work on thinking about how we can use embeddings, how to scale market basket analysis, think about the concept of modules and so on and so forth. Rajan did the annotation. I have two students who were visiting here for about 6 months, 2 years in a row, and did all sort of that grunt work of extending the context. It's very complex, and some collaborators from the University of Delaware who came to us with this question, which was super exciting. And pretty much that's it. I'm happy to take any questions you may have, and sorry I talked the whole hour.